Hi, my name is Judy, and my poster is on the applications of LCMS in clinical diagnosis of tyrosinemia type 1. In LCMS proteomics, complex mixtures of proteins are subjected to enzymatic cleavage and undergo analysis using a mass spectrometer. The steps are as follows: extraction of desired proteins, removal of contaminants, digestion of proteins into peptides, separation into homogeneous peptide mixture, and identification via MS. In order to identify the protein, MSMS is used, and identification occurs via comparison to a database of previously discovered peptides. LC is first used to separate molecules as follows. Peptides of similar molecular mass but different hydrophobicity elute from the LC column and enter the mass spectrometer at different times. Due to the complexity of the proteome, multiple separation steps are employed to spread out the proteins according to different chemical or physical characteristics. This step also helps to avoid molecules of identical molecular masses producing a single intense peak in the MSMS spectrum. ESI is then employed prior to MS analysis. During ESI, the capillary needle is the inlet into the apparatus, and here the liquid is nebulized and charged. A voltage is applied to the sample, and the ejected molecular ions are oxidized upon entering the desolvating capillary. Evaporation from heated gas decreases droplet size, and the electric field density becomes more concentrated. This causes the ions to repel one another, separating positive and negative charges. The fragmented molecular ions then travel to the entrance of the mass analyzer for analysis. QTOFMS is an analytical technique that is used next and functions to combine the benefits of two unique mass analyzers and occurs upon molecular fragmentation. The quadrupole consists of two pairs of parallel metal rods, and voltage is directly applied to the rod pairs. Their potentials are continuously altered, and thus the pairs are out of phase with each other. The MZ ratio of the ions will determine if the ion will either travel the full path of the quadrupole and reach the detector or crash into the rods. The precursor ion can be fragmented through a process known as collision-induced dissociation, which yields important information on the structure of the molecule. The usage of TOF is also necessary as ions with lower MZ will travel the fastest and arrive at the detector first, while ions with larger MZ will travel the slowest. This will narrow the possibilities for potential molecular formulas. LCMS is commonly utilized in biochemical screenings for genetic disorders, such as in the diagnosis of Tyrosinemia type 1. When analyzing the genetic disorder, the analyte of interest is the amino acid tyrosine. The detrimental effects of tyrosinemia type 1 can cause nervous system breakdown and increased risk of liver cancer and kidney failure. Since tyrosinemia type 1 is incurable and lifelong, early detection via LCMS is vital in ensuring the disease does not progress. The usage of LCMS to diagnose tyrosinemia type 1 has mediocre efficiency at first because slightly elevated levels of tyrosine are found in premature or jaundiced babies. Scientists may begin by adding complexity to the LCMS through high throughput and direct injection techniques. Another method to improve tyrosinemia type 1 diagnosis is to test the levels of succinylacetone. This amino acid is a specific marker of tyrosinemia that is not indicative of jaundice or premature birth. Thus, detection of high levels of succinylacetone and tyrosine are indicative of tyrosinemia type 1. The efficiency of LCMS detection of tyrosinemia type 1 holds great impact over the quality of life for individuals with this condition. If treatment is delayed, tyrosinemia levels run rampant, which has long-term effects of liver cancer and kidney failure. However, with early detection via LCMS, individuals with tyrosinemia type 1 can begin symptom management and avoid the extremes of the condition. The impact of LCMS in clinical diagnosis is undeniably relevant, and due to technological advances such as improvements in mass spectrometry sensitivity and chromatographic resolution, it has been proven without a doubt that LCMS-based analysis of metanephrine and normetanephrine is superior to urinary analysis and has led to improvements in steroid analysis. The future of LCMS will explore the integration of NMR to LCMS-based metabolic profiling and metabolic studies. MSMS database will likely increase as the knowledge base of known molecular markers increases. The introduction of informatics tools for automated generation of candidate empirical formulas and stereoisomers may become available, as well as algorithms to predict chemical structure based on fragmentation spectra.